So the preparation of the formulation should be done separate, like the suspension case, drug molecules, and the immersion. Drug molecules will be packaged in the immersion, and the solvent separate, which is oil. So this is a picture of propofol. Looks like what? Milk solution? Yeah, so the immersion drug may look like this. So that depends on the hydrophobicity of your drug. So it may have more aqueous phase, or it may have more hydrophobic oil phase. So it depends how your drug will be formulated. Okay, so it may come in different types. So methods of dispersion range from simple agitation, mixing, to the use of high shear equipment such as collide mills, ultrasonifier, and homogenizer. So dispersion. So it may require different processes during your formulation. That's what it means. Okay. So because your drug molecule may be strongly hydrophobic, sometimes you may wrap that drug molecule with something like liposomes around. So then what is liposome? Liposome, right here, its structure is a hydrated phospholipid vehicle. Vehicle is a delivery kind of molecule, okay, where active ingredient is incorporated into the inner hydrophobic core of the liposome. So here you go. So liposome, it looks like what? It's kind of a cell membrane, right? So if you consider this is a cell, there is a lipid bilayer, right? So just like that. So your drug molecule right here is in there, because maybe your drug molecule is strongly hydrophobic or hydrophilic, depends on what the structure is. So you can package your drug using liposome, okay? Or it could be packaged into something called the micelle as well. Micelle is different than liposome. What's the difference? It's just a lipid single layer versus double layer. So micelle case, see, these are all strong hydrophobic hydrocarbon chains. So your drug molecule may be strongly hydrophobic. Then you may want to wrap your drug molecule with micelle instead of liposome. Liposome, both outside of liposome and inside liposome may be hydrophilic, okay, because your lipid has amphiphilic kind of characteristics. So these has hydrophilic, but these hydrocarbon chains are hydrophobic. So inside here are hydrophobic, but outside or inside this space may be hydrophilic. So your drug molecule may not be strongly hydrophobic if you're going to wrap your drug with the liposome. So there is a difference. Okay. So understand the chemical structure of a liposome, why it's needed. Okay, or micelle, so that they are a little different. Again, so lysosome structure is a three-dimensional structure, of course. Lipid bilayer, so just like that. So it's wrapping your DNA in there. A lot of DNA-based drug molecules. So you probably heard about coronavirus vaccines. Among those different vaccines, so those Moderna vaccine and Pfizer vaccines are made with RNA molecules, and then Johnson & Johnson, and then another company, their vaccine is based on the DNA. But the Moderna vaccine and Pfizer vaccines use I think it's a lipid-based delivery when they formulate the vaccine. So they will use something like that when the vaccine is packaged. Okay. So liposomal formulation can deliver low molecular weight compounds or proteins and peptides, and they are very important in the formulation and delivery of DNA-based therapeutics, like DNA or RNA-based therapeutics. So like gene therapy, for example, normally that gene made up out of DNA molecule is normally packaged with those liposomal formulations and delivered into the patient's body through injection. Okay, Moderna drug, RNA-based drug, was also packaged with liposome and delivered that way as well. Okay, So gene delivery by liposomal system combine cationic and deuterionic lipids. What that means is, going back to the structure, these are cations, okay? So positively charged ion here, okay? So that's possible. So the cation lipid facilitates DNA complexation, while deuterionic lipid helps in membrane perturbation and fusion. So what it means is, again, these are lipid bilayer. So your cell membrane is also lipid bilayer. So this liposome can be fused with the cell membrane. That's what it means. So it can help the delivery of drug molecule into the cell through that mechanism. Okay. The main challenge in liposome formulation is encapsulation of the active ingredient into the liposome matrix. What that means is, how do you insert your active ingredient here? So here inside. So that's what it means. It's a little tough. Okay. And next kind of dispersed drug called microspheres. So they are solid particles that are in micron sizes. Those are microspheres, like spear shape. Okay, those particles have different sizes, from 1 to 1,000 micron sizes, and they are formulated to sustain the release of drug, just like adjuvant in vaccine. Okay, since drug molecules are coating on the surface of these microspheres, they are slowly released from these microspheres. So the whole purpose of coating microspheres with your drug is to have the slow release of the drug, or sustained release of the drug. That's the whole purpose of it. Okay, so they normally serve as, I told you, adjuvant for vaccine. Okay, slow release mechanism is helped by adjuvant in your vaccine delivery system. And then non-suspension drugs, nanosuspensions are dispersed system with nanometer size of insoluble drug particles. So nanometer size are very small. So, although they are not dissolved, okay, but they behave like, kind of a solubilized drug. So that's what it means. So there are, instead of microspheres, nanospheres could be used. That's what it means. Okay. Okay, then we're going to talk about the formulation of freeze-dried powders. Okay, so lyophilized drug powders. Okay, so these are typically the picture of lyophilized drug. So there's no water in there. The water is all freeze-dried. During the time, the water is crystallized and evaporated, removed from your drug. So now you just have dried cake of drug molecules left in your primary container. So that's the freeze-dried drug, what they look like. Okay? So it turns out about 40% of biopharmaceutical drugs are freeze-dried. Okay? So that's a lot of drugs. Okay? Why? Why is it so popular? Because there are benefits of freeze-drying. Okay? So the products can be dried without elevating temperature. So when you dry, you may think, oh, you can remove water if you boil it, right? But if you boil it, then your biological molecule will be denatured, so you don't want that. So instead, you freeze it first and then slowly dry it. So that's why it's called freeze drying process. Okay, so under the condition, water molecules that are crystallized can be vaporized directly. So that's the principle of freeze drying. Okay. So the main benefit of freeze drying is extended stability of your drug. So, the main reason of drug going bad during the shelf life is through the chemical process called hydrolysis, right? So normally water molecules involved in the chemical conversion, we call that hydrolysis. But if you remove water molecules, so hydrolysis would not happen. So you can prevent degradation of the drug molecule through hydrolysis. So you can get an extended shelf life that enables the extended, prolonged stability of your drug products. So that's the main benefit. So although the cost of the freeze drying process, adding that especially into your drug manufacturing, may be expensive, but prolonged shelf life gives you benefit. Then you can freeze dry your drug. Okay. So once you freeze dry it, of course, these cakes should be able to reconstitute it back to the solubilized form before giving the injection. So you normally have to add sterilized UFI back to your drug before giving the injection. When you do that, the drug should be suspended during kind of very easily meaningful time. Within a minute or two, you can easily resuspend it, reconstitute your drug. Okay, so that is a requirement. If you can do that, yes, that's fine. But if you cannot do that, then you cannot freeze dry. So that's another problem. Okay. So when freeze dried product is present in small quantity, it's hard to detect the freeze dried drug. So what it means is, now you can see the cake, white fluffy cake at the bottom of the vial, primary container easily, so you can see if it's freeze dried, right? But if your drug molecule is not visible like this after freeze drying, then it's hard to know if you still have your drug in there or not, right? So to give that visibility, sometimes bulking agent is added in your freeze drying formulation to make these very visible, fluffy, nice looking cake after freeze drying. So that's what this statement means. Bulking agents may be added to make a drug visible after freeze drying. Okay, so one of the most commonly used bulking agent is mannitol. We'll talk about mannitol again. Okay, so freeze drying is the removal of ice, crystallized water, or other frozen solvent from material through the process called sublimation. Okay, understand this term, sublimation. So that's a very important term. Okay, what's sublimation? That's the evaporation of water molecule from crystallized form. So think about three states of water. Okay, water is liquid under room temperature, right? And then it freezes at the freezing point at 0 degrees Celsius, becomes crystal form. Okay, and water liquid can be vaporized if you boil it. So the liquid can become vapor. Okay, then the question is, can you change crystal water form into vapor form without going through liquid form? Okay, so the question for that, the answer is of course yes. You can vaporize crystal water, which is ice, without going through liquid form. That's what sublimation is. Sublimation is. Okay, sublimation means your water is vaporized from water crystal, ice, directly without going through liquid form. Okay, how do you do that? Yeah, so you have to freeze it first. So you have to cool down the temperature, right? So it can be done on the very cold temperature. Then how do you vaporize it? If you apply a vacuum pressure, so low temperature and high pressure condition, then yes, your water crystal can be vaporized directly. Okay, at a low temperature condition and high vapor, the pressure, then yeah, your water can sublimate. Okay, so freeze drying is based on this condition. So low pressure and low temperature condition, your crystallized water will sublimate. Okay, so that's it. So lyophilization and freeze drying are the terms that are interchangeable. Okay, but actually, it's freeze drying is more descriptive term when you try to explain the process, because if you look at this process, you freeze your water first, you drug first, and then you dry it. Okay, you are not freezing and drying at the same time. It's a sequential process. You freeze it first and dry it. So freeze drying is a better way of explaining the situation than lyophilization. Okay, so controlled freeze and then drying will keep the product temperature low enough during the process to avoid change in dried product appearance and characteristics. It's an excellent method for preserving a wide variety of heat-sensitive material. Okay, so that's why you freeze dry. Okay.